All right, cell communication part 3, hopefully the final step of this. Now we are talking about response, okay? The response to this extracellular signal. So ultimately, a signal transduction pathway, which is that whole thing we just mapped out in the last video, what does this do? Leads to the regulation of one or more cellular activities. Leads to the regulation of cellular activities. Right? And many of these regulate protein synthesis. All right, many regulate protein synthesis. Turning specific genes on or off. They can turn genes on and off. Regulate the activity of proteins rather than causing their synthesis. Can also regulate activity of proteins. Or protein activity. Cool. Okay, tell me more. This is all about response. The specificity of cell signaling. There's this whole big figure 11.16, which is just crazy looking. Elaborate enzyme cascades amplify the cell's response to a signal in each catalytic step in the cascade. The number of activated products can be much greater than the preceding step. As a result of the signal's amplification, a small number of epinephrine can lead to millions of epinephrine molecules bonding to receptors on the surface of a liver cell or muscle cell, can lead to the release of hundreds of millions of glucose molecules from glycogen. And we remember that glycogen is a polysaccharide made up of glucose monomers, which is extensively branched. Two cells respond differently to the same signal. There's crosstalk. All right, we have these things called scaffolding proteins. Right, we have scaffolding proteins. And what do these do? Proteins, purple. What does a scaffolding protein do? Large relay proteins to which other relay proteins are simultaneously attached. Increased by the presence of. They increase efficiency. Increase signaling efficiency. Simultaneously binds to a specific activated membrane receptor and 3 kinases. It's basically like bonding. It binds to protein kinases. It binds to these protein kinases. All right, binds. So if we remember, our protein kinases, which are these guys, which I know, well, they're green because they're enzymes, but our protein kinases, let's say we have, 1, 2, 3, our protein kinases. Our scaffolding protein is going to kind of look like this. All right, and these are our protein kinases. Which, remember, are enzymes. That's what a scaffolding protein does. So, in termination of the signal, we did not show the inactivation mechanisms that are a central aspect of a cell signaling pathway. For a cell of a multicellular organism to remain capable of responding to incoming signals, each molecular change in its signaling pathway must last only a short time. So we saw in the Claire example, if a signaling pathway component becomes locked into one state, consequences can be serious. The ability of a cell to review new signals depends on reversibility of the changes produced by prior signals. The binding of signaling molecules to receptors is reversible. As the external concentration of signaling molecules falls, fewer receptors are bound in any given moment. By a variety of means, the relay molecules return to their inactive forms, the GTPase activity. To a fresh signal. Signal response can be terminated quickly because ligand binding is reversible. This thing that started at the very beginning, this ligand, right? We should even write that that is a ligand. Can be terminated. Okay. I mean, again, there's this really scary type of thing here. We have the specificity of cell signaling. I think for the portion of time that we have where this happens, cell transduction pathway leads to the regulation of cell activities. And you know, responses occur in the nucleus or in the cytoplasm. In the nucleus or the cytoplasm. Okay, I don't know, I feel okay about that, about response. Different pathways, regulate protein. So the last part of this chapter is something called, we're going to go right into this. When signaling pathways were first discovered, they were thought to be linear, independent pathways. Our understanding of cellular communication has benefited from the realization that signaling pathways components interact with each other in various ways. For a cell to carry out the appropriate response, cellular proteins must integrate multiple signals. Let's consider an important cellular process. Cellular suicide is an example. Cells that are infected, are damaged, or have reached the end of their functional lifespan often undergo programmed cell death. Best understood type of this is apoptosis, meaning falling off. During this process, cellular agents chop up the DNA and fragment the organelles and other cytoplasmic components. The cell shrinks and becomes a lobed, that's called blebbing, and the cell's parts are packaged up in vesicles that are engulfed and digested by scavengers. Apoptosis protects neighboring cells from damage that would otherwise suffer if a dying cell merely leaked out all of its contents, including many digestive enzymes. Right, so we have this thing called, so we'll go ahead and just kind of cut this off. I'll even leave some space if I end up finding some more information on this. But we're going to go ahead and talk about apoptosis. Which is this. A programmed cell death. A programmed cell death. You know, and you have these scavenger, what are they called? Scavenger cells. Scavenger cells digest its parts. Engulf its parts and digest. Several apoptotic signaling pathways exist in the cells of humans and other mammals, triggered in different ways. Signals eliciting apoptosis can originate from outside or inside the cell. The signal that triggers apoptosis can come from either outside or inside the cell. Outside the cell, signaling molecules released from other cells can initiate a signal transduction pathway that activates the genes and proteins responsible for carrying out cell death. Within a cell whose DNA has been irretrievably damaged, a series of protein-protein interactions can pass along a signal that similarly triggers cell death. Key gateways into appetitic program, relay proteins integrate signals from several different sources. Built-in cell suicide mechanism is essential to development and maintenance in all animals. The similarities between apoptosis genes in the nematodes and mammals. There we go. Ready? When you have too much apoptosis, you have Alzheimer's, right? Too much. Alzheimer's. Right? Too little, and there's not enough, cancer. Interesting. Finally, some medicine, right? All right. If I need to draw more, write more about cell communication, I'll use this. But that is the cell communication chapter. Give an example of apoptosis during embryonic development and explain its function in the developing embryo. Wow. Why would a cell die in embryonic development? Oh. Apoptosis will create your fingers, right? You have your fingers here. That's pretty cool. Well, I mean, this is a paw in a mouse, but if you have this, I mean, that's an awful picture, but eventually you'll have right? And that will kind of be in here. So your apoptosis comes in and kills all this. Oops. That's pretty interesting. Okay. We'll say that's a mouse paw. Cool. Cell communication. Chapter 11. Done.